Dear friends, most welcome to all of you. In the series of video lectures, we have discussed different topics of genetics. Now, we are going to explain one more important concept of genetics. This is polygenic interaction. To understand the basics of topic, first of all, let us recapitulate monogenic inheritance. Then we can contrast and compare mono and polygenic inheritance for better understanding. Now let us start. Now, as we know that monogenic inheritance is that type in which one character is taken at a time. Suppose we take example of character height in pea plant. It has two traits, tall and dwarf, which acts as a parent, one male, another female. We can use genes, for example, capital T, small t for tall and dwarf respectively. We can use these terms as a gametes formed from tall and dwarf. Now these gametes later on fuse to form first generation of seeds. This is called F1 generation, or we can say filial first generation, that is capital T, small t, heterozygous tall. We can interbreed F1 individuals, or we can say selfing, and we get F2 individuals or F2 generation. The F2 generation formed is capital T homozygous tall, capital T small t 2 times heterozygous tall, small t, this will be the dwarf. This is F2 generation. Now here we get phenotypic ratio 3:1 as shown in diagram, 1:2:1 genotypic ratio. From this cross we can conclude some important points. In monogenic inheritance, one gene control one character. Very important point. And its father is Gregor John Mendel. Now see towards the diagram. Here the F1 individual resembles with one of the parent, that is tall. Here the tall parent is dominant parent. Now we can add very important point to the topic. Dear friends, listen very carefully. The capital T forms tall. Presence of number of capital T also forms tall, not taller. So we can say quality of dominant capital T matters, not quantity. Hence monogenic inheritance is also known as qualitative inheritance. Hope you got the concept. See towards F2 generation. Here total tall parents are 3 and 1 is dwarf. So we can write here that total amount of dominant parent is 75%. Recessive parents are only 25% in the form of dwarf. Here we can write that no intermediates are formed in monogenic inheritance. The graphical representation of monogenic inheritance shows straight line. That is, when we are going to plot a graph between tall 75% of individuals and dwarf 25%, we get straight line graph. Dear friends, you know in a monogenic inheritance only one gene is involved which controls one character. Hence we can write here that environment has very less influence on single character, single gene. So we can write here the monogenic inheritance get less influence from environmental factors. Now last not least very important point of monogenic inheritance is that in monogenic inheritance two extreme phenotypes are formed. One is tall, another is dwarf without intermediate formation. So this is the case of discontinuous variation. I hope you got the concept by all means. Now coming back to our main topic, that is polygenic inheritance. Let's proceed. Polygenic inheritance is also called multigenic inheritance. Here, do not compare polygenic inheritance with multiple alleles. They are totally different. We can clear this confusion in a separate video bite. Now talking about polygenic inheritance, this term was first coined by Mather. In polygenic inheritance, many genes control one character in an organism. Dear friends, the father of polygenic inheritance is considered as a Carl Reuter. He discovered first polygenic inheritance in corolla length in tobacco flower. In polygenic inheritance, the F1 generation formed are all intermediate phenotypes. They do not resemble with any parent. Now, when we get F2 generation in polygenic inheritance, the F2 individual phenotype depends upon number of dominant alleles, means quantity of dominant alleles. So we can say the person having one dominant allele is different from the person having two dominant alleles. Hence, this polygenic inheritance is also called quantitative, cumulative, or additive inheritance. Hope you got the concept. Now coming to the very important point about polygenic inheritance, that is, the polygenes are also known as multiple genes. Here two types of alleles used. For example, dominant alleles used are known as contributing alleles. They play a significant role to control phenotype. Recessive alleles are known as non-contributing alleles. The environmental influence is maximum in polygenic inheritance because many genes get exposed to the environmental conditions. Now we have very important point. In polygenic inheritance, two extreme phenotypes are not formed, but phenotypes are formed in numbers by continuously showing minute changes due to addition of dominant alleles. Hence, this is the case of continuous variation. Now, if we are going to plot a graph in polygenic inheritance, we get bell-shaped curve. Look towards this graphical representation. It shows high peak at the middle as intermediate phenotypes. Dear friends, we have prominent example of polygenic inheritance. Now let's explain. That is skin color in human beings. It was first studied by Davenport in 1913. According to Davenport, skin color in human beings is controlled by three polygenes or multigenes, namely A, B, and C. Now let me show you how these genes interact and control skin color. Let us crossing Negro and albino individuals. These act as parents and this will be the genotype. Negro acts as a dominant parent, albino acts as a recessive parent. The color is controlled by three important genes. Now, Negro will form gametes like this, albino gametes will be formed like this. These two gametes fuse, one acts as a sperm, another egg, it will form first generation, or we can say F1 generation. This F1 generation offsprings, they are known as mulattoes and intermediate. Now we can interbreed mulatto F1 individuals to get F2 generation. Let me show you total gametes from F1 individual. They are 8 in number. After crossing two mulatto individuals, we get F2 generation in polygenic inheritance, that is in skin color of man. Here we get total phenotypes equivalent to 64. These 64 individuals as a phenotypes are in the ratio of 1:6:15, 1:20, 1:15, 1:6:1. They are total 64. Now let me write their names. Negro 1, accordingly 6 are very dark in color. We have 15 phenotypes which are basically dark in skin color, and 20 they are mulatto or intermediates. Again 15 they are light in skin color. Accordingly 6 are very light in skin color, and there is one individual, it is albino. Now let me show you contributing alleles or dominant alleles involved in these phenotypes. Now proceeding from Negro to albino phenotype, we have dominant allele number in the form of 6, 5, 4, 3, 2, 1 and 0 respectively. Means 0 dominant alleles are present in albino. Now let me show you some good examples here. We have genotype, for example, capital A, capital A, capital B, capital B, capital C, capital C. Here total dominant alleles are 6 as mentioned in the above chart. It will be Negro simply. Now we can quote one more example. We have one genotype like this. You can count total dominant alleles are about 4. So from above chart, this individual possesses dark color in skin. Accordingly, you can write number of genotypes and calculate its phenotype. Dear friends, it was all about polygenic inheritance topic. I hope you got the concept by all means. If you find this video valuable, kindly share. Never forget to subscribe. Thanks. Have a nice day.